So next up, got, I believe, Chantal Vogels from the Yale School of Public Health, who will also be speaking about their Amplicon-based sequencing method that they've developed. So, Chantal, you should be able to share your screen.
Yep, thank you. Let's see if I can pull this up. All right, can you all see that all right?
Yes.
Okay, good. So thank you for the introduction. My name is Chantal Vogels. I'm an associate research scientist at the Yale School of Public Health. And in my talk today, I'm also going to be talking about the Amplicon-based sequencing approach that we developed.
Just a quick thing, you might want to put your slides in presentation mode.
Oh, they are. Is that looking better now?
Perfect.
Okay, great. Sorry about that. So I want to start by just giving a quick introduction of PrimalSeq, which is the tool that we use to develop our primer scheme. So PrimalSeq was initially developed by Josh Quick and his colleagues during the Zika epidemic. Back then, it was really hard to get good coverage when sequencing Zika virus from clinical samples. So they came up with this tool to basically amplify the entire genome by designing overlapping primer sets. So this has been used a lot during the Zika virus epidemic, then was adapted during the SARS-CoV-2 pandemic, kind of like similar ID, 400 base pair amplicons, slightly larger genome this time. And then with monkeypox, we're dealing with a slightly different beast here. So it's a double-stranded DNA genome, much larger. And that's why we decided to generate a primer scheme with larger amplicons. Back when we started this work, we basically did not have that many genomes available. And that's why we decided to pick an older genome from the UK in 2018 to develop our primer scheme. So our scheme consists of about 163 primer pairs, and again, an amplicon size of about 2,000 base pairs. And the whole idea behind our approach is that you can plug it into existing SARS-CoV-2 amplicon-based sequencing workflows. Because especially during the pandemic, so many labs have invested in genomic infrastructure, and we really wanted to utilize that beyond SARS-CoV-2. Slightly different though, like given that it's a double-stranded DNA virus, there's no need for the cDNA synthesis steps, so those can be skipped. And when we were validating our primer scheme with the Illumina COVID-seq protocol, we just had to change the final cleanup step to get rid of both the small fragments and the larger fragments by basically just changing it to a double-sided cleanup. For bioinformatics, there's many different pipelines that labs are using. Key here is that you update your reference genome and the associated bed files. The bed file basically just contains the primer position so that you can accurately trim the primers of your reads. And with some pipelines, you'll have to update the genome length. I'm not going to go into too much detail here because the next presentation is from Kevin Libet from Teogen Genomics, and he's going to dive into the bioinformatics pipeline that they developed for monkeypox. So when we developed this primer scheme, we reached out to our colleagues at the Massachusetts Department of Public Health. So Glenn Gallagher and his team really helped us to do the initial validation, where they were already doing metagenomic sequencing on the clinical samples that they got. And they were able to actually do this pairwise comparison of the Amplicon-based approach with metagenomic sequencing on the same 10 samples. So this is really the work of Luc Gagné and Matt Doucette with help from Danny Park, where they basically sequence these samples in parallel. I'll spend a little bit of time to explain this graph because I'll be using kind of like the same style of graph throughout my presentation. On the x-axis, you'll see the CT value. And with higher CT values corresponding to a lower viral DNA load and lower CT values corresponding to a higher viral DNA load. And then on the y-axis, we show the percent genome coverage at 10x. So basically at least 10 map reads at each position. And you can see that if you have a lot of DNA in your sample, both methods basically perform really well. So you can get a good coverage, but you will see that there's a quick drop-off for coverage that you can achieve with metagenomics, especially at these higher CT values, while you're still able to get decent coverage with the Amplicon-based approach. So after this initial result, we immediately made our protocol publicly available so that other labs could try it as well. And basically what we did is we generated a bunch of aliquots. This is really the work of Nick Chen, who's been pipetting for hours and hours to prep lots of aliquots so that we could send these out to other labs. And so far, we've been sending primer aliquots to more than 35 labs. We asked these labs, like, hey, if you're starting sequencing, would you be able to share some of your data with us so that we can basically expand on this validation study? So far, we've had 12 labs that have shared their data. Most of them are public health labs in the US, but we also had a couple of international collaborators in the UK, in Portugal, Vitor, that we heard of his presentation earlier today, and in Brazil. These labs have been using two different sequencing technologies, both Illumina and Oxford Nanopore technology sequencing. And we have been able to test it with two different sample types, so both lesion swabs and oropharyngeal swabs. So all these labs did the sequencing, they shared their data with us, and then we basically streamlined the backend bioinformatics so that we could determine coverage on the same way for all these different labs. And we used IVAR for that. So the first set of graphs that I'm going to be showing is labs that generated their data with Illumina, and they used lesion swabs for sequencing. And you will see that all these 8 labs were successful in getting good coverage across a range of CT values. So basically, again, the same graph with CT value on the x-axis and the % coverage on the y-axis. You see that for CT values up to an average of around 28, labs are able to get good high coverage. We used polynomial regression to get a sense for that CT threshold where we start to see the drop-off, which turned out to be around a CT of 28. Some labs had a narrower range of CT values, and that's why you see that the line is not really dropping off yet. But again, consistently across these labs, we get good coverage. Then in this next,
79, right?
Yeah.
In this next set of
I'm gonna go around.
We have 2 labs that actually were able to sequence from both lesion swabs and oropharyngeal swabs. The number of oropharyngeal swabs was rather low, so we have not really been able to determine any thresholds with this. But this is really just to illustrate that you can actually also generate good coverage out of these oropharyngeal swabs. Interestingly, the lab in Massachusetts, the Massachusetts Public Health Lab, was able to distinguish between oropharyngeal swabs where no lesions were present in the throat and swabs where lesions were present. And in general, there is a difference in CTs, but we show that for either, we are able to get good coverage. At INSA, there were a couple samples that from the lesion swabs that performed a little bit less well. We think that this is probably due to some technical error, not necessarily the primer scheme. Then the next comparison I want to show is labs that were able to sequence on Oxford Nanopore technology sequencers, and they basically use the SARS-CoV-2 workflows that Arctic has developed to plug these primers in. So at NHS Lothian, they were able to do a pairs comparison. So they both sequenced the same samples on Illumina and on ONT. And basically you see that you get roughly like the same results, so very comparable genome coverage between both platforms. We saw a little bit more variation in the data from Johns Hopkins, but again, it's probably like some other factors that come into play here. But in general, we kind of show that both platforms are able to get good results. And that really gets back to that point of like, we really want people to use what they're already using and not kind of like reinventing the wheel. Given that we saw some variation in coverage between the labs, we were kind of interesting to see like, okay, we see this clear effect of CT value, but what does the number of sequencing reads do in terms of coverage? So for this plot, we took our data that we generated in our lab. We did that on the NovaSeq, so we were able to generate a lot of reads. We then downsampled reads per sample so that we could actually get a sense for the difference in coverage as a result of
I haven't been listening to this one. I was like, we have to go to our other meeting if so.
So if we compare 500,000 reads to 2 million reads, we can actually see a difference in coverage of about 0 to 26%. Especially at higher CTs, we see a bigger difference. We then also wanted to see, okay, so what should we really target in terms of the number of reads that labs should generate? And there we see that basically there's about a 2% difference if you sequence or if you generate 1 million versus 2 million reads per sample. And that really suggests that up to that threshold of about a CT of 28, we recommend to target about 1 million reads. Where do you start seeing the gaps? If you either have high CT values or if you have lower sequencing reads, well, that's what we try to show with this graph here. The yellow indicates a coverage below 10. So we see that high CTs, basically you start seeing a lot of gaps, which makes sense because you're really not having enough DNA there. And you also see that effect of a higher number of reads where you get like a better coverage. Consistently, there's two amplicons where we see lower coverage, and that's amplicon 75 and 188. This is not due to primer mismatches. And I feel like it's similar to what Matthijs was presenting just before the break. Like, there's probably just differences in PCR efficiency, and perhaps that can be overcome by changing some of these primer concentrations. Our study has some limitations. We are targeting the clade 2B. If you were to try to sequence viruses from other clades, I would expect that you would get a lower coverage, simply because these genomes are going to be more divergent. Also, in this last graph, what we show is that the primer efficiency is not optimally balanced between all these 163 primers, which, again, makes kind of sense because it's a big primer scheme, and you would expect some differences there. But again, overall, we get really good really good results consistent across all the labs that have been doing the validation. This was mentioned already a couple of times earlier today. So inverted terminal repeats are very challenging. It can lead to multiple primer binding sites, and it can create some challenges when you're mapping your reads. So that's really something we need to keep an eye on. And hopefully by either masking or, paying attention to this, we can make sure that doesn't cause any issues. And then lastly, also already mentioned earlier, these orthopox viruses are quite notorious for genomic rearrangements. So I think Amplicon-based sequencing can really help to increase the throughput of sequencing, but we recommend that periodic long-read metagenomics would be a good strategy along with that. I have to say, though, that our collaborators at the Los Angeles County, they were actually able to detect a 600 nucleotide deletion, and with our approach, that is now affecting the qPCR assays. And so it is to say that, you might be able to detect some of these genomic rearrangements like deletions, but it won't always work. So to conclude, I think that Amplicon-based sequencing provides a cost-effective and widely applicable alternative to metagenomic sequencing for human monkeypox virus surveillance. Based on our validation study, we kind of came up with 3 different recommendations. One, really utilize existing Amplicon-based sequencing infrastructure. Like, we don't try to reinvent the wheel, but we really want to utilize what's already there. Especially now that so many labs can do Amplicon-based sequencing. We would say prioritize samples with a CT below 28, especially if your resources are limited. And if you do want to try sequencing higher CTs, you might get partial genomes, but you might lose some coverage there. And then along with that, we suggest to generate at least 1 million reads per library to make optimal use of your resources. I want to end by thanking all the amazing labs that we've collaborated with. Like, it's really them who did all this work, sequencing, sharing validation data, and it's really been a great collaboration between both academic and public health labs. I also want to say some special thanks to Nick Chen, Crispin Jaguza, Luc Gagne, and Glenn Gallagher, who've been co-leading this study. And with that, I'm happy to take any questions.